Biology 101, Lecture 15. Hello, students. Today we're going to take a step back and look at populations and how they come up with these interesting ideas. I have no idea. But here we go. We're going to start looking at populations and what their problems are and how they all work. So, microevolution is a change in allele frequencies in a population over generations. So, that means that in a population, there can be very, very small changes, and they may be carried over. And what we're looking for is the rate of change over generations. Three mechanisms can cause allele frequency change. One is natural selection, another is genetic drift, and the last is gene flow. Only natural selection causes adaptive evolution. Variation in heritable traits is a prerequisite for evolution. Genetic variation among individuals is caused by differences in genes or other DNA segments. And this genetic variation can be measured as gene variability, or in reality, it's really nucleotide variability, isn't it? Natural selection can only act on variation with a genetic component. A mutation, as we all know, is a random change in nucleotide sequence of the DNA. A point mutation is actually a change in just one base in a gene. Chromosomal mutations that delete, disrupt, or rearrange many loci are typically pretty harmful. Duplication of small pieces of DNA increases genome size, and that's usually less harmful than the deletion of DNA. Mutation rates are low in animals and plants. It's pretty good. The fidelity, as we know, is pretty high. But the average is about one mutation in every 100,000 genes per generation. Mutation rates are often lower in prokaryotes and higher in viruses, and that's because viruses duplicate pretty rapidly when they do. Mutations accumulate quickly in prokaryotes and viruses because it's due to the short generation times. Sexual reproduction can shuffle existing alleles into new combinations, which is more important than mutations, and we looked at gene shuffling a few lectures back. So before we even talk about populations, we should actually define it. A population is a localized group of individuals capable of interbreeding and producing fertile offspring. A gene pool consists of all the alleles for all loci in a population. So that is the real definition of a gene pool. A locus is fixed if all individuals in a population are homozygous for the same allele. If there are two or more alleles for a locus, diploid individuals may be either homozygous or heterozygous. And that brings us to the Hardy-Weinberg principle. The Hardy-Weinberg principle is actually really set for an ideal population. And of course, in life that isn't really true, but we always like to conceptualize something which is idealized or a model, something to refer to, so we can see how far away from perfection are we. The Hardy-Weinberg principle looks at an ideal population where no evolution is occurring, and the allele and the genotype frequencies stay constant generation after generation. And we know that never happens. However, in reality, there's always genetic drift, there's always gene flow, there are always mutations, and there are always other factors in populations, so we can never reach Hardy-Weinberg equilibrium. We shouldn't. The frequency of an allele in a population is calculated by multiplying the total number of individuals by 2. That gives us the total number of alleles at a locus. And the total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual, plus 1 allele for each heterozygous individual. And it's the same calculation for recessive alleles. By convention, if there are 2 alleles at a locus, we don't use A and B or any other alphabet. We just use P and Q to represent their frequencies. the frequencies of all alleles in a population will all add up to 1, right? So for example, P plus Q is going to equal 1. For example, let's consider a population of wildflowers that is incompletely dominant for color. And we have a population consisting of 320 red flowers, and they're dominant red, 160 pink flowers, so they're heterozygous, and 20 white flowers, and they're homozygous recessive. If we calculate the number of copies of each allele, this is how we would do it. So for the red, which is dominant, for one copy, we're going to take 320, because that's how many flowers we have red, right? 320 times 2, because there were 2 alleles at each locus. So 320 times 2. But then we can't forget the pink flowers. They also have 1 allele. So we add on 160 to that, and we get 800. Now, if we should find the calculation of the white flowers. So there are 20 white flowers multiplied by 2, because it's recessive and it's homozygous. And then, of course, we look at the pink flowers, and sure enough, there are 160 pink flowers that have 1 locus of that allele, and that's 20 times 2 plus 160, which is equal to 200. So to calculate the frequency of each allele, this is how we would do it. For P, for the frequency of red, we take 800, which is what we figured out from before, from the previous slide. That's how many instances of that allele are. And then we divide it by 800 plus the other version of the same gene. So that would be the white. So 800 plus 200 is equal to 1,000. 800 divided by 1,000 gives you 0.8. And then we'd calculate the frequency of the white allele 2. That would be Q. And that's 200 divided by 800 plus 200, which gives us 0.2. And so the sum of the alleles is always equal to 1, which is 0.8 plus 0.2, which is 1. If we select alleles at random from a gene pool, so this is how we would do it. The big tub has red balls and white balls. We're going to say that they're the alleles for the red flowers and the white flowers. And we know that the frequency of the of P is 0.8 and of Q is 0.2. So when we have gametes that are produced, the chance of each egg producing red is 80 and white is 20. And same thing for the sperm. They will produce 80% red and 20% white. So if the observed genetic makeup of the population differs from expectations, under Hardy-Weinberg, it suggests that the population is evolving. So Hardy-Weinberg will always say that the population is not evolving. And when we actually go look at a population and we get numbers that are different, we will say that the population is actually evolving, because we'd compare it to the Hardy-Weinberg ideal. If a population has gametes randomly contributing to the next generation and Mendelian inheritance occurs, allele and genotype frequencies remain constant from generation to generation, then the population is indeed in Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium describes the constant frequency of alleles in a gene pool. Consider, for example, the same population of 500 wildflowers and 1,000 alleles, where P was equal to 0.8 and Q was equal to 0.2. Then the frequency of the genotypes can be simply calculated like this. For homozygous dominant red, that's P squared, we would just square 0.8 and we get 0.64. For heterozygous, which was going to be 2PQ, we would just multiply the answer, the numbers, and we get 0.32. And for homozygous recessive white flowers, it's Q squared. So we'd square 0.2 and we get 0.04. And if we add it all up, it'll come up to 1. The frequencies of genotypes can be confirmed using a Punnett square. like this. So 80% red with 20% white. You get sperm made, 80% are going to have the red allele and 20% will have the white. Same thing with the eggs. And as you can see, when you cross them over, you indeed do get the same idea, which Hardy-Weinberg's equation does. You just don't have to use a big clunky Punnett square, and you can use just a one-line formula or a binomial. So here it shows you 64% homozygous dominant red, 32% heterozygous pink, and 4% recessive homozygous white. So the gametes of this generation with random mating, these gametes will result in the same mix of genotypes in the next generation, so it doesn't matter. This population is therefore in Hardy-Weinberg equilibrium. If P and Q represent the relative frequencies of the only two possible alleles in a population at a particular locus, then using the binomial P squared plus 2PQ plus Q squared will equal to 1, where P squared and Q squared represent the frequencies of the homozygous genotypes, and 2PQ represents the frequencies of the heterozygous genotype. The Hardy-Weinberg theorem describes a hypothetical population that is not evolving. In real populations, allele and genotype frequencies do change over time. The 5 conditions for non-evolving populations are rarely met in nature. First, there should be no mutations. Right away, you know that that's not going to happen. Second, there should be random mating. Well, mating is usually selective. It isn't random. Number 3, no natural selection. So that is also a big problem because the pressures of succeeding in the environmental niche that organism lives in is indeed natural selection, and the ones that are not very successful will just be weeded out. Or if the population is extremely large, then I guess that would be a condition. Or if there is no gene flow, and that also is difficult. The differential success in reproduction results in certain alleles being passed to the next generation in greater proportions. 3 major factors alter allele frequencies and bring about most evolutionary change. So natural selection, genetic drift, and gene flow. And we're going to look at these one by one. Genetic drift and gene flow do not consistently lead to adaptive evolution, as they can increase or decrease the match between an organism and its environment. The effects of genetic drift, let's see, what are they? 1. Genetic drift is significant, but only in small populations. Genetic drift can also cause allele frequencies to change at random. And genetic drift can lead to a loss of genetic variation within populations. And genetic drift can also cause harmful alleles to become fixed. So if we look at this, this picture, genetic drift is occurring here. So what is going on? We have the same red, pink, and white flowers, in generation 1. Here the frequency of the red is 0.7 and the whites are 0.3. But 5 plants die off or leave the pool. They just disappear. Maybe somebody bought them. So if they selectively picked red ones, then the what will happen is now the generation is now changed, and P, the frequency of the red, is no longer 0.7, it's 0.5, and Q has now raised itself to 0.5 also. So if we again take out 2 more plants, we will have another generation, and let's say we just take out all the white plants. We will get a population that is going to end up being all red. And so here in genetic drift, we have actually eliminated something that was maybe desirable. Maybe not, but we certainly have eliminated some variety. There is another effect that occurs in populations, and that's called the founder effect. The founder effect occurs when a few individuals become isolated from a larger population. And that's why they're called founders. The allele frequencies in the small founder population can be different from those in the larger parent population. So they just go and cut off and go live, like, say, in an island or something like that. There's also something called the bottleneck effect, and that will also change populations. So there's a sudden reduction in population size due to a change in environment. And the resulting gene pool may no longer be reflective of the original population's gene pool. If the population remains small, it may further be affected by genetic drift. So you're compounding the problem. This is how a bottleneck effect would actually, it really is a bottleneck. So you stuff them all in, nothing much comes out, and you have very little surviving population. This will make the new population, the little population, very, very different from the original population. Gene flow consists of the movement of alleles among populations. Alleles can be transferred through the movement of fertile individuals, or gametes, like pollen, and gene flow tends to reduce variation among populations over time. Gene flow can decrease or increase the fitness of a population. There are three modes of selection. There's directional selection, which favors individual at one extreme end of the phenotypic range. There's disruptive selection, which favors individuals at both extremes of the phenotypic range. And then there's stabilizing selection, which favors intermediate variants and acts against the extreme phenotypes. Evolution by natural selection involves both chance and sorting. New genetic variations arise by chance, and the beneficial alleles are sorted and favored by natural selection. Only natural selection consistently increases the frequencies of alleles that provide reproductive advantage. So if we look over here, in this picture, what we have is an original population of little mice, and it seems like one has very light color and one has a very dark color, and there are three that have medium-ish light color, but varying type. All of a sudden, in directional selection, what will happen is one extreme is just cut out, like the white one, and so we have the original population and then the evolved population in the red line. So it has shifted. In disruptive selection, what we did was we only favored the two extremes and got rid of all the nice medium brown mice. So we end up with a very different population from the original one. And that's why the graph looks like two red humps. Or if we look at the stabilizing selection, again, we're changing the population because we got rid of the white ones, we got rid of the dark brown ones, and we got rid of, anything that wasn't quite smack dab in the middle. So this would change the frequencies of alleles a lot. Striking adaptations have arisen by natural selection. For example, certain octopuses can change color rapidly for camouflage. For example, the jaws of snakes allow them to swallow prey larger than their heads. So here we're seeing a great big old snake, and he's chomping on this enormously big egg, which is bigger than his eyes are bigger than his mouth. No, he can actually stretch his jaws because the bones are actually movable. The hinge is movable. We don't have that. We have a fixed hinge in our mouths at the jaw bone, whereas the snake actually has a movable jaw. Natural selection increases the frequencies of alleles that enhance survival and reproduction. Adaptive evolution occurs as the match between a species and its environment increases. Since the environment changes, adaptive evolution is a continuous process. Genetic drift and gene flow do not consistently lead to adaptive evolution, as they can increase or decrease the match between an organism and its environment, as we just saw in the previous pictures with the mouse. Natural selection cannot, however, fashion perfect organisms, because selection can only act on existing variations, and adaptations are often compromises, and chance, natural selection, and the environment all interact together. Goodbye, students.